Stevia is 300 times sweeter than sugar but has 0 calories. Scientists finally know why. Researchers at the Chinese Academy of Sciences just decoded the exact genetic switches that let this plant manufacture molecules our taste buds mistake for pure sweetness. Drop a stevia leaf on your tongue and sweetness floods your receptors within seconds. No glucose, no fructose, no metabolic cost. The stevia plant, Stevia rebaudiana, native to Paraguay, produces compounds called steviol glycosides that are 200 to 300 times sweeter than table sugar. Your T1R2 and T1R3 taste receptors, the same proteins that detect sucrose, bind to these molecules with equal enthusiasm. But unlike sugar, steviol glycosides pass through your digestive tract unmetabolized. 0 calories, 0 insulin spike, full sweetness delivery. The FDA approved purified stevia extracts in 2008, and global stevia production now exceeds 8,000 metric tons annually. The plant looks unremarkable: small white flowers, green serrated leaves, but inside those cells, a genetic assembly line is rewriting the rules of sweet. So how does a plant build a molecule that hijacks mammalian taste? In 2024, a team led by researcher Zheng Jianming at the Chinese Academy of Sciences mapped the complete stevia genome and pinpointed the exact gene cluster responsible. The plant uses 3 key enzymes, UGT76G1, UGT85C2, and UGT74G1, to attach glucose molecules onto a backbone called steviol. Think of it like Lego bricks snapping onto a core. The first enzyme attaches one glucose, the second adds another, and the third locks on a third sugar unit. This cascade produces rebaudioside A, the most potent sweet compound in stevia. The researchers discovered that a single transcription factor gene, SRMYB, acts as the master switch, ramping enzyme production up or down. Knock out SRMYB in lab plants and sweetness drops by 80%. Overexpress it, and the plant floods leaves with rebaudioside A at triple normal levels. But the genome reveals something chemists didn't predict. Not all stevia tastes the same. Wild stevia plants produce 11 different steviol glycosides, each with a slightly different glucose arrangement. Some, like stevioside, taste bitter alongside the sweetness. Others, like rebaudioside M, are purely sweet with 0 aftertaste. The genetic map shows that environmental stress flips which enzymes dominate. This discovery is rewriting agriculture and medicine. Armed with the full genetic map, researchers are now engineering stevia strains with single compound output, plants that make only rebaudioside M, the clean sweet molecule. Stevia's 300 times sweetness isn't magic. It's a 3-enzyme assembly line controlled by a single master gene, and scientists just handed us the blueprint. The plant your ancestors chewed in Paraguay is now a genetic template for 0-calorie sugar factories. If you want the next breakdown of biology rewriting food, subscribe to @BrainyWizBros and catch tomorrow's mechanism. Subscribe to @BrainyWizBros for next breakdown.